Hi, this is Mr. Anderson, and today we're talking Punnett squares. And on your screen you see a picture of Reginald Punnett, and he, with another scientist slash mathematician named William Bateson, came up with this idea that genetics and math are linked after they had gone through the work of Gregor Mendel. And so they took Gregor Mendel's work and figured out how we could demonstrate it mathematically. And in fact, what you're looking at on the screen is called a monohybrid cross. So a monohybrid cross is where we're looking at one gene with two possible alleles. And we refer to that situation as Mendelian genetics or Mendelian trait. So this shows the possible inheritance of a gene that has two possible alleles. And so I'm going to show you in a minute how we set it up using this square that's on your picture. But before I do that, I want to also recognize that we start looking at Mendelian genetics because they're the easiest to understand. But in real life, in the actual world, almost all the human traits, the majority anyway, are called multifactorial traits or complex traits. And what that means is that most traits for a human are actually caused by the interaction of several genes. So most human traits are not Mendelian. There are a couple that are, and as we discover more and more medicine, we discover that there are some diseases that are Mendelian in nature too. So we'll highlight a couple of those as we go through our unit together. So let's start going through some of the information that's happening in this square. So it looks pretty simple, but there's actually a lot going on, a lot that's been tied in here. And first, let's start with Mendel's law of segregation. So Mendel was a guy ahead of his time. He realized in his work with pea plants that when we have a gene with two possible alleles, even though he didn't know what it was, he understood the underlying condition that this thing we call today meiosis was happening. And remember in meiosis that each parent has two copies of a chromosome. Right? So in a human, that means that if we're talking, let's say, for example, one parent could have the dominant allele, that would be for, let's say, brown-colored eyes, and another parent might have the recessive trait for blue-colored eyes. Well, you're not going to get both of these traits from a parent. Only one of these alleles will be passed on because you only get one chromosome from your parent. And so on our Punnett square, the first thing to recognize is that there are two sides. One side's for the father. Generally, we set the father at the top. And so we have one possibility. In his sperm cell, we could get either big B, or we could get a sperm cell that passes along a little b for blue eyes. I do want to recognize that I am oversimplifying eye color. Eye color, we believe, is a multifactorial trait, and I'm presenting it as a Mendelian trait just for the example. I also want to refresh you on the fact that we call these sex cells or gametes with the sperm from the father, or when we're talking the mother we usually put on the side of the Punnett square. And her possible alleles, remember these alleles are different versions of a gene that would come from a chromosome here. So her possible alleles Let's say that she has blue colored eyes. So that means that she would have one little b on one chromosome and another little b for blue eyes on the other chromosome. So both of her alleles are the same. Both of the father's alleles are not the same. We have a big B and a little b. And that's one of the reasons why we call these homologous chromosomes, meaning that they're similar, but they're not identical. Right? Remember, that's the point of meiosis. It recombines the genes during crossing over, and it creates genetic variety. So let's put another egg cell in for the mother, and then let's move her chromosomes over here with those possible egg cells that they would get passed on in. Okay, so let's take a look of how to actually set up this Punnett square now that we know all this background information. So let's look at an example that we saw from the Amoeba sisters before with PTC tasting. Remember that this is a recessive trait where you could taste, the recessive version of this trait don't taste a bitterness in some types of vegetables related to like broccoli, Brussels sprouts. People who have a dominant version of the gene, remember dominant versions of genes are shown as capitals, do taste bitterness, meaning when they put PTC paper in their mouth, they sense this horrible bitterness. And non-tasters are recessive, meaning they have to get 2 copies of this little b allele. Okay, so let's put that away for a second and let's set up our problem here. So, parents that are both heterozygous for PTC tasting have a child. What trait will their child have? So we need to use the terms here and break them down. Remember, hetero means different. So if the parents are heterozygous, that means both parents, this is the father. Let's go ahead and make a little box for him showing this is the father. He's heterozygous, so he has one big B and one little b. And the mother is also heterozygous, so she has a big B and a possible egg cell, or a little b and a possible egg cell. We don't really know which is going to get passed on. So we have to create this Punnett square to show both possibilities for each parent. So when we do these, the father's genes will get moved down to each box. And let me show that here. So when we are moving the alleles in these examples, the father's allele will travel in the first box and it will also travel in the second box. And when I'm looking at my setup here, I'm going to actually put his allele over here because we usually make the dominant allele first in the box. It doesn't matter in the overall scheme of things, but it helps when you're trying to read it later. Alright, so in the next box over here for the father, he has a little b, and his little b allele will travel down here, and then let's put another arrow, and his little b allele will also travel down to the bottom box. Okay. So that's how I got the b's in those boxes. Now let me get rid of these arrows. And we're going to do the same thing with the mother's alleles. So the mother's big B allele is going to travel to this box. And her allele will also travel to the second box. So over here I'll also have a big B. And then the same thing happens down here. But in this case, we have a little b. So I'll have little b. And likewise, her first allele will travel to the first box. So now I'm going to move my arrows out of the way, and we can see that we have 4 genotypes. Remember, genotypes is a gene combination. So let's point out before we move on that these letters represent alleles, possible versions of a gene that you inherit from the parent, and these gene combinations together are the genotypes. So big B, little that's a genotype. Little b, that's a genotype. And big B, that's a genotype. Let's take a look at another type of problem. So this problem is cystic fibrosis. This is a condition that affects cells that make up the lungs. People who have cystic fibrosis have cells that create a sticky mucus that makes it difficult for them to be able to breathe well, and it gets trapped in their lungs and can cause lots of infections that can actually be life-threatening. So this is a recessive condition, and we're told that one parent is homozygous recessive for cystic fibrosis, and the other is homozygous dominant for normal lung cells. We don't really know which parent is which here, so it doesn't matter. We're going to move little f over here. This would be our homozygous recessive. This is the parent. In this case, we're making it the father who has cystic fibrosis because it's at the top. And we'll say the mother is homozygous dominant, meaning she has normal lung cells. So I'm going to start this one. Since mom's homozygous dominant, I'm going to bring her alleles across first. So we put a little f here, and then we go over to the next box, and we put a little f over there. We'll do the same thing in the bottom box. We'll put a little f, a big F, a capital F, there for mom, and the same thing in the next box as we bring her allele over. Now we'll bring the father's alleles down. And so in the first box, we'll have a little f. The box below, we have a little f. Go back up to the top, we have a little f. And in this box at the bottom, a little f. So the question was, what is the chance their child will have cystic fibrosis? To figure this out, we have to read the allele combinations. We have big F, little f for every possible situation. So we have to use our knowledge now of dominance and recessive to figure this out. And since every possible gene combination the child would inherit has a dominant allele for normal lung cells, this child would have normal lung cells. The child, him or herself, would be heterozygous, meaning they carry one copy of each of the alleles for this gene. The gene would be lung cells. Okay, let's put this together, and I'm going to show you a more formal way we write this in our last one. So our last one is widow's peak, and a mother is heterozygous for a widow's peak. And the father has a straight hairline. And if you don't know what I'm talking about, I included a picture from Utah Genetics on the right. So in the picture you can see that somebody who has a widow's peak has this kind of triangular shaped hairline in the front. And if it's a straight hairline, well, that's just straight across. So a mother is heterozygous. And I want to show you another trick here. So sometimes when we're doing these, we wind up with letters that are hard to tell apart when they're dominant or recessive. So I showed how I tell the difference personally. I made one capital, as we always do for a dominant, but the difference is for my recessive allele, I underline it. And that's a trick I use to help myself keep them organized. when the letters are really close, I underline the lowercase one, so I can tell that it is lowercase for sure. So mom's, oh, I'm putting that the wrong spot. Let's fix that. So mom is heterozygous. Remember, mom's generally shown on the left. That's what I almost made a mistake with. And the father has a straight hairline. So that means that he has to have 2 copies of this recessive allele. So let's make another copy of that and move it into place here. And now we can set up our Punnett square. So we know that mom's first allele is going to go over to this box, big W, and then that allele will also go over to the next box. And down here, mom's other allele is a lowercase w, so I'm going to underline that so I can tell it apart. And I'm actually going to just copy that, because that allele will also travel over to my next box. Okay, now dad's alleles are both recessive, so this allele will travel down this box, and the allele will also travel down to this box. And likewise, the same thing on the other side. The allele travels down to the first box and then the second. And I think I'm going to put a little space. Oh, I don't like that. We'll fix that. I'll just leave it as it is, I guess. So now we can look at the results for this gene combination. The last part to show you is called the genotype and phenotype ratios. And that's where we break down the possibilities from the genetic combination. So in this question, it asks, what type of hairline will their 3 children have? So to figure this out, we have to first write the genotypes. Remember, those are the gene combinations we could have. So one gene combination is big W, little w, and the other is little w, little w. We need to also write the ratio here. So we have 2 of these are big W, little w, and 2 of them are little w, little w. So we could write this a couple of ways. We could write 2 big W, little w, to 2 little w, little w. Or you could use your math skills for reduction. Since 2 and 2 could reduce, right, they could reduce to 1, this could be a 1-to-1 ratio also. The other thing we have to do is we have to write the phenotype. Okay. So in this situation, let me make a new box. So remember that phenotype is the physical trait. The physical trait here is whether we're talking straight hairline or widow's peak. And widow's peak is a dominant trait. So big W, little w would give us widow's peak. So we have 2 widow's peak. To little w is straight hairline. So I'm going to make this box a little bigger. To 2 straight hairline. Clean up my grammar a little bit here. All right. So, or my punctuation. There we go. We have 2 widow's peak to 2 straight hairline. Now this would also be 1-to-1, or we could say 50% widow's peak to 50% hairline if we were supposed to write it in percentages instead of a ratio. Okay, so either way, that's our phenotype outcome. Okay, so let's address the question we've been asked. What type of hairline will their 3 children have? Well, we don't have a good answer because Each child, every time parents have an offspring, a child, they're going to have the same chance. They're going to have 50% chance of inheriting big W, little w, or 50% chance of inheriting little w, little w. Each time. So the first child has 50% chance of inheriting either of these genotypes, which means they have 50% chance of inheriting either one of these phenotypes, widow's peak or straight hairline. The second child has the same 50% chance of inheriting the same gene combinations and the same phenotype. The third child has the same 50% chance of inheriting each gene combination and each phenotype. So, all we can tell those parents is their child each time has 50% chance of having either straight hair or a widow's peak, which I'm sure they wouldn't find to be a very helpful answer. Okay, so I want you to go through and try the practice problems in your assignment. And there is a solutions video that will show you half of the answers at the end. That way you can check your work and see if you're doing it correctly before you turn it in. Thanks, and until next time, take care.